Okay, hello to all of you. Let's discuss chapter number 14, part 2. And in this part, I'm going to discuss about the non-Mendelian genetics. So in part 1, we discussed about the Mendelian genetics. Now we're going to talk about the non-Mendelian genetics. So give me 1 second. Okay. Okay. So we are going to talk about the inheritance patterns which do not follow the Mendel's law. The very first example I'm going to talk about is incomplete dominance, and then I'm going to talk about codominance. So the inheritance patterns in which the complete dominance is not followed in some of the genetic patterns, some of the genetic inheritance patterns, the law of incomplete dominance is followed, in which in the heterozygous situation, the dominant alleles do not dominate completely, and recessive alleles are not expressed at all. That doesn't take place. That was in the case of Mendel's inheritance pattern, where the law of dominance was followed, in which one of the allele was a dominant allele, another allele was a recessive allele. The dominant alleles express themselves, and the recessive alleles they do not express themselves in the presence of the dominant alleles. The recessive alleles express themselves only when there is no dominant allele there. So the recessive alleles can express themselves only in the absence of the dominant allele. So that is the case of Mendelian genetics, in which law of dominance is followed. So this is an inheritance pattern in which the law of incomplete dominance is followed. So in which if there is a pure red plant with a red flower, and then there is a plant with white flower, so when they are going to cross in the heterozygous situation, it is not going to be it's not going to be plant with the red flowers, but it's going to be a plant with the pink flowers. So that is incomplete dominance. And when the plants from this generation, they are crossed in the homozygous situation, the plant will be having red flowers, and again homozygous situation, the plant will have white flowers. But in the heterozygous, if the Mendel's law was followed, if the Mendel's law of dominance was followed, the flowers would have been red. But in this case, where the law of incomplete dominance is followed, the flowers are going to be pink in color. Next, I'm going to talk about codominance. Okay. Now, there are some genes which can have more than 2 alleles. So till now, we have studied all genes have 2 alleles. Like one is on paternal chromosome, another is on maternal chromosome. In the diploid, in the diploid organisms, of course, there are 2 chromosomes, but there can be more than 2 alleles. So out of these three alleles, there can be only 2 alleles which can be chosen. Now, for example, the typical example of codominance is the blood type. So we know that there are 4 blood types, right? There's A blood type, B blood type, AB, or O. So the blood type A, B, and O, basically why somebody has a blood type, why somebody has B, or somebody has AB, it is all on the basis of some antigens on the surface of the red blood cells, and those antigens are nothing but they are glycoproteins. Basically, they are basically proteins with some carbohydrates on them. So there are so for this blood typing, we have when I say the blood typing, means for the expression of these carbohydrate antigens or the glycoproteins on the surface of the red blood cell, there are basically 3 alleles involved. One is A, another is, so one is IA, IB, and this is small I. So this one codes for the antigen A, this one codes for the antigen B, and this allele codes for none of the antigen. Okay, so if this allele is there, the antigen is going to be A. If this allele is there, the antigen is going to be B. And if this allele is there, it is going to be neither A nor B. So you can see here, this is the genotype. If the genotype is, if both the alleles are capital A, the blood type is going to be A, and it is going to show these antigens on the surface of the red blood cells. If one of the antigen is A, one of the allele is capital A, another allele is small I, which means it is not going to be coding for any antigen. The blood type is still going to be A, because this does not code for any antigen. So this one codes for antigen A. So you can see these are the red blood cells with these kind of antigens. Now this, if both the alleles are capital B, the blood type is going to be B, and these are the B antigens on the surface of the red blood cells. If one allele is capital B and other is small I, again the blood type is going to be B. If one is A, one is B, the blood type is going to be AB. And if both the alleles are small i's, it means no expression of any antigen. This blood type is going to be O. So for the expression of these antigens on the red blood cells, there are 3 alleles. Okay, and this is now the blood type AB. The blood type AB is a typical example of codominance, because here the antigen A and antigen B both are expressing in the presence of each other. So this is codominance. Okay, so none of the allele is dominating, no allele is dominating. So one allele is not dominating the other allele. So both alleles can express themselves in the presence of each other. So none of the allele is dominant allele, and none of them is the recessive allele. That's what it means. So this is codominance. Okay, here again Mendel's law is not followed. Now pleiotropy, some of the genes, they have multiple phenotypic effects. So that situation is called pleiotropy, or those genes are called pleiotropic genes. For example, pleiotropic alleles are responsible for multiple symptoms of hereditary disease like cystic fibrosis and sickle cell and anemia. And then we have, now we're going to talk about what is epistasis and what is polygenetic inheritance. So epistasis is that inheritance pattern in which one gene affects the phenotype of another. So there are two genes. There are two genes, and both genes form some products. Those products interact with each other, and then they influence the expression of some character. So for the expression of some character, if two genes are involved, they influence the expression of each other. That is called epistasis. And in polygenetic inheritance, multiple genes are involved which affect a single trait. Polygenetic inheritance, multiple genes independently affecting a single trait. Epistasis, one gene affects the phenotype of the another due to the interaction of their gene products. So epistasis, basically, I am going to explain you with the help of an example. So basically, here is the example of these dogs. Okay, so you can see that this is a dog with black fur. Okay, so the color of this coat of the fur of the dog, it is actually dependent on two genes. For example, in Labrador retrievers and many other mammals, coat color depends on two genes. Okay, so one gene determines the pigment color. If the allele is capital B, the color of the coat is going to be black. If the allele is small b, it's going to be brown. If the allele is capital B, it's black. If the allele is small b, it's brown. And the other gene which is involved is E. If the allele is capital, with the alleles E for color and small e for no color. So basically the other gene which has two alleles, capital E and small e, if the allele is capital E, which means that pigment which is expressed by this gene, okay, if the allele is capital E, it means that the pigment will be deposited. And which gene? So this is another gene which is determining the color of the pigment. If the allele is capital B, the color is black. If the allele is small b, the color is brown. But this pigment will be deposited only if the other gene which is involved has allele capital E, because if capital E is there, then this pigment will be deposited in the hair, and if the allele is small e, then the pigment will not be deposited. So you can see here, now this is a dog with capital B, small b, capital E, small e. So it has capital B, which means the color is going to be black, and because this second gene has allele capital E, it means that this pigment will be deposited, so the dog will have color. So this is basically a cross, and this is a little modification of the Mendel's dihybrid cross. So you can see here, so this is a cross, and it's an example of epistasis. It shows that how one character is governed by, or it is affected by two genes. So this is a cross between these two dogs, and you can see these are the gametes formed, and you can see this is the Punnett scare, and this is the progeny which is possible. So you can see this is a dog with capital B, capital B, capital E, capital. So both things are there. This is going to show black color. This is also having both. So both. Let's talk about this. Now this dog has both the alleles small b, which means the color is going to be brown, and because this has both capital E's, the second gene has both alleles capital E. This pigment is going to be deposited, and this dog is brown in color. Now if you look here, although both of the alleles are capital B, which means this allele is coding for which color? Black color. But because for the second gene, the allele is small e, which means no pigment. Pigment will not be deposited. So in this case, if this allele is small e, this pigment, whether it's black or brown, is not going to be deposited, and the dog will have this kind of a color. And same here. There is one allele for black color, but these are both small e's, no color. And this one is both alleles are small b's. Both alleles are small b's, and these are also small e's. So no deposition of the color. So no color. No, sorry. This small b is for brown color. Small b is from brown color, but because both alleles are small e, so this dog is not going to have any color, either black or brown. So this is epistasis, and the ratio here is the modification of Mendel's dihybrid cross. Okay, what is polygenetic inheritance? Polygenetic inheritance is that inheritance pattern of some characters like skin color or the height, where more than one gene, more than two genes are involved. An additive effect of two or more genes on a single phenotype. Example, height. Over 180 genes affect height. We are talking about genes. We are not talking about alleles here. We are talking about genes. 180 different genes are involved in the height. How much height or what kind of height a person will have, it is dependent on the effect of 180 genes. Similarly, skin color also. And this is basically a cross showing that how three different genes A, B, C. I mean, basically this is just showing the skin color. This is just an example showing that how three genes are involved, but there are more than three genes, and you can see how the gametes are formed, and you can see how the different shades of the skin are possible. So this is polygenetic inheritance. The last one is multifactorial. So multifactorial is when the expression of some character is not only dependent on the genotype, but it is also dependent on the environmental factors. So traits that depend on multiple genes combined with environmental influences are called, inheritance pattern is called multifactorial. Like you can see, these are some flowers. If you grow them in the basic soil, if the pH is alkaline, they're going to give red flowers. And same plant gives blue flowers if it is grown in the acidic soil. So this inheritance pattern is called multifactorial, in which not only genes are involved, but the effect of environment is also there. So it's an interplay of genes and the environment. So that's all, chapter number 14 part 2.